I'm Megan Greenfield and I'll be talking about animal tissues. Stratified epithelial tissues are found anywhere that friction may occur, for example in the skin. It covers the exterior body surfaces. It's made up of multiple layers known as stratified squamous, stratified cuboidal, and pseudostratified columnar. One layer is in contact with the base membrane, but the others stick to one another to maintain structural integrity. These are used as a second layer of protection to prevent pathogens getting through. Simple epithelial tissues are closely compacted and found in places such as your kidneys, lungs, etc. They are used for secretion and absorption in the small ducts of glands and kidney tubules. The upper surface is exposed to the outside of the body and the basal surface rests on the connective tissues. Connective tissue splits into 5 sections. Dense regular tissues are wrapped around muscle, cartilage, nerves, ligaments, tendons and bones. They are densely packed collagen fibres arranged in parallel and they are very strong. They form strong rope-like structures like tendons and ligaments. Dense, irregular tissues are found in the dermis of the skin and in the walls of large tubular organs. They are disordered and have a mixed formation made up of a dense weave of thick collagen fibres. Their function is to effectively bind different various tissues together to form organs and to act as a protection. This is split into areolar, adipose and reticular. Areola are found in most body locales beneath epithelial tissues and they are a meshwork of collagen fibres. Their function is to attach epithelial cells to the underlying tissues and to allow for diffusion of nutrients to epithelial cells. Adipose tissues are found on the skin and around vital organs. They are loose fibrous connective tissues packed with adipocytes to store fats. They store lipids and insulate and cushion organs. Reticular tissues are found in the lymph nodes and in the spleen and liver. They are found to be in a series of branching threads and the reticular fibres that are present are fragile and form the stroma. Their function is to form supportive framework for the soft organs. Supporting connective tissues are found surrounding blood vessels and around internal organs. They are strong and durable tissues and consist of dispersed cells that lack intercellular contact. Their function is to form the structural framework for the body, transport nutrients and waste through the body, and to provide protection for vital organs. Fluid connective tissues are found in the tissue spaces. They are the matrix of the tissue and there are various types of organic materials in the form of colloid function. Their function is to transport nutrients and gases around the body and defend the body against pathogens. Motor neurons are found in the central nervous system and they assist other neurons by carrying electrical impulses from the CNS to the rest of the body. It has a long axon which receives and sends messages and it protects the outside of the spinal cord and controls the effect to organs. Sensory neurons are located underneath the skin and part of the peripheral nervous system. It transmits sensory information such as light, temperature, etc. to the brain so it can process it. It has a smaller axon as the messages it sends are smaller. Both of the neurons send messages in one direction towards the central nervous system and they both start at the dendrites and end at the terminal knobs. But the sensory neurons work mainly in the peripheral nervous system whereas the motor neurons work in both the peripheral and the central nervous system. Also, the sensory neuron triggers the brain to process data, but the motor neurons send electrical impulses to muscles or glands. In addition, the sensory neurons pick up the stimuli, but the motor neurons take their reactive message away from the CNS. Action potential is the short-lasting event where the electrical membrane potential of a cell quickly rises and falls. It follows a consistent movement of the path, which is the axon. The salutatory conduction of action potential is an electrical impulse which helps the action potential to move with efficiency and speed. The whole act is carried out within the myelin nerve fibres. It comes in 2 forms which are the excitatory and the inhibitory. Muscle tissues are split into cardiac, smooth and skeletal muscle. The cardiac muscles are found in the heart and are involuntary, meaning they have to work to keep us alive. They are needed for blood and haemoglobin distribution so it doesn't fatigue. They are very thick and dense and to prevent fatigue, they have a good blood supply and a large number of mitochondria to respire and to have a large ATP supply. Smooth muscle tissues are not striated and contract in a peristaltic wave structure. The function of this is to maintain important body functions, move food and waste through the intestine and to move the urine through the bladder. Skeletal muscle is striated as the microfibrils run parallel to each other. They are attached to the muscle and then to the bone to allow movement and found all around the skeleton. The function is to protect the skeleton and to cushion and support us. Also, it gives structure for movement. The muscle is voluntary, meaning it doesn't have to work. There are 2 types of twitch muscles, fast and slow. Fast twitch muscles are needed for a short time and these are used in sports like sprinting. Slow twitch muscles are needed for endurance and for longer times and used in marathon running. The function of these is dependent on the muscle as to whether it's fast or slow twitch. The sliding filament theory involves muscles. Muscles are made up of long individual muscle fibres, which are made up of myofibrils. These are then made up of sarcomeres, which contain thick and thin filaments. Thick filaments are made up of myosin molecules, and thin filaments are made up of actin molecules. The contractions of muscles is generated by the thick and thin filaments sliding past each other. The contraction happens by binding by the myosin head with actin molecules, propelling the actin towards the M line. The power stroke is the key step generating the movement upon phosphate binding. That is it on animal tissues. Thank you for listening.